Hello, everybody, and welcome back. Thank you for attending session 2, which is on advancing the use and interpretation of meaningful within-person change thresholds. A reminder and a couple of housekeeping items. So all attendee lines will be muted during this. The workshop is being recorded and will be available on our website by the end of May. In addition, we will have a question and answer session. And at that point, you'll be able to raise your hand and we'll call on you for your turn to ask questions. I'm having a little delay right now on advancing the slides, so bear with me for a second here. Yeah, so T, I don't seem to be able to move the slides forward. So, okay, we've got our raise hand slide here again. So let's move on, see if we can get, okay, all right. So disclaimers, as stated before, the views and opinions expressed in the following slides are those of the individual presenters and should not be attributed to their respective organizations or companies, the US FDA or CPAP. These slides are the intellectual property of the individual presenters and protected under copyright laws of the United States of America and other countries, used by permission, all rights reserved, all trademarks are the property of their respective owners. So the session objectives. The first presentation is going to describe the setting international standards in analyzing patient-reported outcomes and quality of life endpoints in cancer clinical trials, Innovative Medicines Initiative Consortium, and the work they're doing on clinical meaningful change. The second presentation will provide a case study from a pro-consortium member's use of the FDA qualified non-small cell lung cancer symptom assessment questionnaire in a phase 3 clinical trial and the evaluation of measurement properties and meaningful within-person change thresholds. In our third presentation, we'll discuss a mixed methods approach to ascertaining a meaningful within-person change threshold of the worst itching intensity numerical rating scale to support a labeling claim for the treatment of chronic kidney disease-associated pruritus. So I will ask the other presenters and panelists to join me at this time in turning on their cameras for introductions. I am Rebecca Speck, Clinical Outcome Assessment Scientist in our Clinical Outcome Assessment Program at CPAP. Our presenters include Carla Mamolo, Director of Health Economics and Outcomes Research at Pfizer, Johannes Giesinger, Assistant Professor, the Medical University of Innsbruck, Josephine Norquist, Executive Director, Patient-Centered Endpoints and Strategy Lead at Merck, and Margaret Vernon, Senior Vice President, General Manager at Evidera. We have 2 panelists joining us from the FDA today. They include Selena Daniels, Clinical Outcome Assessment Team Leader in the Division of Clinical Outcome Assessment, and Lily Gerard, Lead Mathematical Statistician, Division of Biometrics 3, Office of Biostatistics, Office of Translational Sciences within the Center for Drug Evaluation and Research. All right, at this time, presenters can join me in turning off their videos, and I will turn the presentation over to Carla.
Hello, thank you, Bex. It's my pleasure to be here today to speak to you about Cyclical, which is the setting international standards for analyzing patient-reported outcomes and quality of life endpoints in cancer clinical trials. And in particular, the work Johannes and I are doing on Work Package 6, which will look at at the harmonization of the terminology and definitions being used for clinically meaningful change. Oops. For our I'm sorry, went too far. For our presentation today, I'll start by giving an overview of the Cyclical project, what led to its formation, our overall objectives, and the work package structure. And then we'll go into Work Package 6 and our work regarding recommendations for clinically meaningful change. So we'll talk about our objectives, the work we did last year, which was Sysaqual's first full year of operations, and also look ahead to what we're doing this year and in the next few years of Sysaqual. So Sysaqual is an innovative medicines initiative. That means it's a joint public-private partnership that receives funding from the European Union and also the European Federation of Pharmaceutical Industries and Associations. And as you can see on this slide, it really is a very diverse initiative. It was really important that we bring together different people, stakeholders, representatives from many countries around the world, but also representing a number of different organizations. We're fortunate to have regulatory agencies involved, including FDA, some health technology assessment groups, patient advocacy representatives, a number of industry partners, several academic groups, and also subject matter experts. And CPATH is one of them. Now, the reason it was so important for us to have all these different stakeholders involved was because Sysaqual was formed to address the lack of consistency and consensus regarding the standards for interpreting, reporting, analyzing patient-reported outcome endpoints, particularly in the cancer literature. So this article here was describes like that early foundational work that led to Sysaqual's formation, and to the project being formed with the IMI. So we have a few overarching goals that we aim to accomplish over the next 4 years. So very importantly is to bring together all these different stakeholders, different perspectives, so that we can discuss, debate, and then ideally gain alignment on developing recommendation statements that can be used to describe the optimal use of PRO data in cancer clinical trials. So that's probably our main objective, is to achieve this international consensus so that these statements can be used and applied by people in different roles and organizations. The aim is also to improve the quality of statistical analysis and reporting in cancer clinical trials. And, the aim would be that this would lead to more consistent and reliable interpretation of this data, and ultimately better use of patient-reported outcomes data for multiple different purposes. So whether it's to support regulatory approvals, health technology assessments, or even shared decision-making between patients and their physicians. So that is our broad goal with this project. So Sysaqual, we divided the work into 8 different work packages. The ones in the orange box represent what we call scientific work packages. So one of them, work package 2, is focused on randomized controlled trials. So they'll develop the recommendations related to PRO analyses in RCTs. Work package 3 is focused on single-arm trials, which are quite common in the oncology setting. And Johannes and I co-lead work package 6, and our focus is on clinically meaningful change. So in addition to this, we also have another work package that is looking at how to communicate PRO findings, how to visualize and present the data. Another work package will look at pilot testing and validation of the recommendations, so that what we recommend is actually practical and workable once you put it into practice. And then we also have work package 8. I'll just call that out because they focus on patient engagement, so making sure we have patient involvement in all the activities that we do at Sysaqual. And I'll just point out that although our scope is focused in oncology, that was part of our the grant we have from the IMI, and that's the scope we're keeping it within. We believe that many of the statements that will be developed should be applicable across different disease areas. All right, so now I'll turn it over to Johannes, who will give you the deeper look into what we're doing on Work Package 6.
So I would like to describe the work within Work Package 6 that will develop recommendations for the terminology surrounding clinically meaningful change and how to define it in the context of cancer clinical trials. In detail, the objectives will be like a harmonization of terminology, which is currently quite diverse when it comes to clinically meaningful change. And we're going to differentiate clearly between group level changes and differences and patient individual level changes and differences. We're going to match these terminologies and definitions to the recommendations developed in Work Package 2 for comparative trials and in Work Package 3 for statistical analysis of single-arm trials. And within Work Package 6, we're also going to identify examples for best practice of the use of clinically meaningful change thresholds in the context of cancer clinical trials. To reach these objectives, we have like 5 tasks within our work package, comprising discussions with a range of stakeholders, 2 types of literature reviews, one focusing on the clinical trial literature and one focusing on the methodological literature, which is mostly about establishing clinically meaningful change thresholds for specific PRO instruments. In task 6.4, which is currently ongoing, we're going to draft proposed statements, the basis for further recommendations on how to use clinically meaningful change in the context of defining endpoints in cancer clinical trials. And this will lead to task 6.5, the actual international consensus recommendations on the terminology for clinically meaningful change and the application of this concept in the context of cancer clinical trials. We have by now completed the literature reviews, and on the following slides, I would like to give a quick overview on some of the findings for the comparative trials we analyzed. So our review relies on previous work done by Madeline P. and published in Lancet Oncology in 2018. And we included additionally publications and protocols provided by Cyclical IMI consortium members. In total, we would analyze 36 trial publications and 22 protocols, all of them making reference to clinically meaningful change in one way or the other. Here, a basic overview on the directions of change of the PRO domains. And we would note that in 60% of the trials and protocols analyzed in this review, there was no specific hypothesis on improvement or deterioration for a specific PRO domain. In about 28% of the trials, there was a direct hypothesis focusing on deterioration of a specific PRO domain, and only 9% this hypothesis would focus on improvement. Looking at the various types of statistical analysis of PRO endpoints, we found that in about half of the trials, the analysis would be based on magnitude of change. That is mostly like mean change or mean difference. This could be like change over time within groups, differences between groups cross-sectionally, or differences between groups with regarding change over time, basically group by time interaction. And as you will notice, the numbers here don't sum up to 100%. This is because like a single trial could use like different methods for analyzing the data. In about 40%, the analysis was focusing on time to event, mostly time to deterioration, that is deterioration for a specific PRO domain. And in 1 out of 6 trials, the analysis was focus on treatment responders, that is an improvement on a certain PRO domain by a predefined threshold. Looking at the application now, how clinically meaningful change thresholds are being used or have been used. This could be like at patient level in 28% of the trials, for group level data in 33% of the trials, and for both in 28%. And it's important to note that in some of these trials, quite a few actually, the same threshold value was used for patient level data and group level data. So the application at the specific level here does not necessarily indicate it has been used correctly. What else did we find? As I said, our review comprised only publications and protocols that would make reference to CMC thresholds in some way, but still only half of these publications would actually use the CMC thresholds for interpretation of the trial results. And even more rarely, like only in 7% of the trials, CMC thresholds were used for sample size estimation for power analysis, which of course also reflects that PRO endpoints are most commonly secondary endpoints of the trials and thus not used for power analysis. So regarding the findings on terminology surrounding CMC thresholds, we found that there's a wide variety of different terms and many of these used interchangeably, although it's not always clear if that's the case. Here, a list of the various terms we identified, and we actually found a difference between the methodological literature and the clinical trial literature in the way that in the methodological literature, the most common term was minimal important difference or change, whereas in the clinical trial literature, the most common term was clinically meaningful change or difference. So this is the basis for us to harmonize terminology within our work package. And when doing this, we're gonna acknowledge the differences between patient and group level CMC thresholds. We're gonna acknowledge differences between within group change or change over time, differences between groups, and differences in change over time. We probably also need to reflect the magnitude of the thresholds, if this is like a lower bound for clinical relevance, or might reflect a clinical relevant difference of a larger magnitude. And probably also going to reflect on the underlying methodology for establishing the thresholds. This might be distribution-based methods, qualitative methods, or anchor-based methods. And finally, we're going to come up with a definition of the meaning of clinically meaningful change. Next to terminology, we're going to develop recommendations on how to use this concept for the various trial settings, various types of statistical analysis. Right now, this table is empty, but we are currently working on making recommendations for each cell in this table, basically for the superiority endpoints, for non-inferiority endpoints, for example, for analysis focusing on mean change over time, magnitude of change, for responder type analyses or time to event analysis. And of course, we're also going to distinguish between comparative trials and the use of the CMC concept in single-arm trials. So the next steps until the end of the project in 2024 will be like the ongoing consensus process for developing the actual recommendations. This will be done in technical language, but also in plain language to allow for the involvement of patients and patient advocates and other groups. All of the recommendations we're going to develop will be pilot tested in real trial data sets to see if it's really feasible and practical, what we're going to recommend. The recommendations for clinically meaningful change thresholds will also be incorporated in recommendations on how to visualize and communicate PRO results from clinical trials. And this will all result in a final publications with an overview of these various recommendations for the use of clinically meaningful change thresholds in the context of cancer clinical trials. With this outlook, I would like to thank for your attention and hand over to the next presenter, Josephine Norquist.
Great, thank you so much. So during this next part of the presentation, I will cover some results on the measurement properties and estimation of clinical meaningful change for one of the qualified instruments from the PRO Consortium, the non-small cell lung cancer assessment questionnaire. In general, I will start with a high-level overview of some of the key milestones related to the measure itself, but also milestones to inclusion of this instrument into one of our trials. And this is the phase 3 Keynote 598. I will give a brief overview of the study design and then specifically the expectation or the evaluation of psychometric properties and clinically meaningful thresholds for the non-small cell lung cancer SAQ. I'll briefly give an overview of the results and the conclusion from these analyses, and then I will share with you some of the lessons learned after completing this study and this activity. So I am here today presenting on behalf of a larger group of great collaborators. So we, within Merck, I wanted to recognize Tom and Eamon, who were part of not only the study team, but also the dissemination team. So we were involved in the study design, collection, interpretation of the data, development of the psychometric analysis plan, and then critically contributing to the report and the manuscript writing. And I'll do a little plug here. All the work that is presented today on the slide has already been published as of April 1st. So the paper is available for all of you to get further details on this work. I also want to recognize our colleagues at IQVIA, Paul and Christina, who helped us finalize the psychometric analysis plan. They conducted the analyses and also drafted and helped us finalize the report and the manuscript. And I also want to recognize our CPATH PRO consortium colleagues, Sonia, Stephen, and Bex, who were part of the dissemination team. And they provided to us the initial psychometric analysis outline and then helped us interpret the results and critically contributed to the manuscript as well. So overall, it was a wonderful collaboration across these three different groups. So on this slide is a brief outline of some of the major milestones that we reached over time regarding this instrument. In November of 2016, within Merck, we decided to include the non-small cell lung cancer SAQ into one of our phase 3 trial, Keynote 598. And I will describe a little bit about the study design. And the intent was to include it to provide additional information on the psychometric property of the instrument and estimating within person change. In March of 2017, the qualification package for the instrument was submitted to FDA by the PRO Consortium. And in April of 2018, the FDA qualified the instrument. And I will give a little bit more details in the next slide about this qualification. In September of 2020, within Keynote 598, we reached the interim analysis 1 database lock. And that's when we used the pooled data across treatment arm to conduct analyses based on a pre-specified statistical analysis plan. In June of 2021, we were able to create a summary report, and this report was shared with the PRO Consortium based on the term of our agreement. And fast forward to pretty much today, the results, as I said, have been published in the Journal of Thoracic Oncology Clinical and Research Reports. So on this slide, briefly can see how it takes several years, from when we and Merck decided to include this measure in our trial to actually having published data. So a brief overview of the instrument itself. Here's the conceptual framework of the non-small cell lung cancer SAQ. It is a 7-item instrument that covers areas of cough, pain, dyspnea, fatigue, and appetite. The items are scored from 0 to 5. The total score goes from 0 to 20. And and the context of use is for adult stage 3B or 4 non-small cell lung cancer that either are treatment naive or have already been treated. And again, this work has already been published and the reference is below. So I won't spend too much time on this part since this information is available for all of you as well. Now, as I mentioned early in 2018, the instrument was qualified by the FDA. The qualification supported this measure as a patient-reported measure of symptom in advanced non-small cell lung cancer for drug development. However, one thing that the FDA clarified was that further evaluation was needed to assess longitudinal measurement properties and to determine what could be considered a meaningful within-person change score. So that's when we included the instrument into our phase 3 study, and here is a schematic of the study design. So on the left-hand side, you can see our key eligibility criteria. So, stage 4 non-small cell lung cancer, no prior systemic therapy, ECOG performance 0-1, and so forth. Each of the patients were randomized 1-to-1 to either pembrolizumab plus ipilimumab or pembrolizumab monotherapy. On the bottom part on the right, you can see the dual primary endpoints, overall survival, progression-free survival, and the list of secondary endpoints. In regards to the non-small cell lung cancer SAQ, this was included as an exploratory endpoint. And as I mentioned earlier, for the objectives were to assess the clinically meaningful thresholds and to conduct psychometric analyses as well. There were also other PRO measures that were included in the study. There were the EORTC, QOQC30, and LC13. And also we had two anchors to help us estimate the clinical meaningful thresholds. And these were the patient global impression of severity lung cancer and patient global impression of change lung cancer. And I'll have the wording in just a little bit. The PRO assessment schedule was determined based on the need of the study. What PROs were assessed at baseline, at each of 3-week treatment cycles until week 18, or what was considered cycle 7, and then less frequently afterwards. One thing to clarify was that the non-small cell lung cancer SAQ, the PGIS and PGIC were administered up to week 18, and I will talk a little bit about this later as well. The analyses were conducted on the PRO full analysis set, and at the end we had 560 patients at the interim analysis 1. We had a cross-sectional analyses were conducted using baseline and week 18, and then the longitudinal analyses were conducted using change from baseline to week 18 or cycle 7. So if we think about the one of the main things that we're talking about today is estimating clinically meaningful thresholds. The primary method was based on using mean change scores at week 18 based on the PGIS at week 18. And the intent was to establish an improvement and a deterioration thresholds. We also used supportive methods, and these were the ECDIFs and PDF curves, and also effect size and standardized response means. The wording on the questionnaire is here on the right-hand side for the PGIS. How would you rate your symptoms of your lung cancer at this time? And one thing that with the 5 response categories, and one thing that I wanted to highlight was that you see here it says version 2.0. We actually started the study with a different version of the PGIS with different response categories, and these were updated after receiving feedback from the FDA. Also here on the bottom of the screen, you see the PGIC wording. However, the primary method was used with the PGIS to estimate meaningful changes. So let's go a little bit in the results. So here on this table are the baseline characteristics of the PRO FAS population. As I said, we had 560 patients, mean age 64, 30% female. And you see the distribution of the ECOG performance status. And at the bottom of the screen are the 2 versions of the PGIS with the different response categories. And just one thing I wanted to highlight is that for this population, we really didn't have a lot of severe and very severe patients at baseline. We conducted a series of psychometric evaluations, and in the next 2 slides is a very high-level overview of them. In general, they all met the thresholds that we had pre-established or pre-specified in our statistical analysis plan. So we looked at item response distributions. There was some ceiling effect indicating the presence of low symptom severity. We had inner item associations, which ranged, as you see here on the left-hand side, from very low 0.1 to 0.82. The internal consistency reliability was above 0.70, as we had pre-specified, and the test-retest reliability was between week 0, cycle 1, and cycle 2, which was week 3, and participants who reported no change was 0.79. So again, unacceptable test-retest reliability coefficient. We also assess different levels of validity, construct validity, convergent and divergent validity. And again, these were all through Spearman correlations with the other measures included in the trial. And these were all moderate to high, so 0.4 or greater than 0.6. We conducted known group validity with the data that we also had available, and again we saw, that we had higher, more severe or lower, worse health-related cutoff depending on the direction of the scores of the other measures that were included in the trial. And we also assessed a level of responsiveness for change from baseline to week 18. And again, we saw that we did have statistically significant mean changes during this time for the SAQ total score based on the PGIS response categories. And we also calculated the effect size and standardized response means, which were in the moderate to large range. So overall, the psychometric properties for the instruments were all met based on the pre-specified criteria that we had in our psychometric analysis plan. Now, when it comes to the interpretation of meaningful within-person change, here's one table that I wanted to present today. And these are the distribution of non-small cell lung cancer SAQ total scores. And the title here should actually see PGIS, not PGIC. These are PGIS category responses. So, and then we have on the right-hand side, the mean and 95% confidence intervals and median and interquartile change for all of these different response categories. So for those from baseline to week 18 who had no change, or those who had 1, 2, or greater than 2 response category changes. And if we look a little bit in more detail, if we look at the category of 1 level response improvement, which is indicated here by the -1, we see that 2.65, a median of 2.65, where a median of 2.25 could be considered the smallest estimate derived from a 1 level PGIS response category improvement. If we go to like the next level, we can see that these numbers go from -5.5 to -5.9 improvement, which could be considered the smallest estimate derived from a 2 level PGIS response category improvement. So one of the things that we have in our paper now is that maybe the recommended threshold could be a range of 3 to 5 points improvement on the 0 to 20 total score range. We also created, in this case, I'm presenting the Ecdf plots. This is Ecdf plots at week 18, which showed a nice separation between the 3 curves. We have the one further to the right, to the left, indicating improvement, no change, and deterioration. So again, a really nice separation on these curves. So we can say that maybe the threshold of 3 to 5 improvement is supported by mean and median change scores for a group of participants who improved, had a small improvement or small to moderate improvement, and it excludes the majority of those who reported no change. Now, one limitation of the study was we were not able to come up with thresholds for worsening. There was a very small sample of participants that worsened, and the variance was large that we weren't able to make a recommendation for a threshold for worsening. Now, these are, some of the estimates. Obviously, sponsors, if they're using this instrument, should definitely engage with the review division early to have these conversations and determine, what threshold might be best used to support a labeling claim for their specific drug development program. So the conclusion from this study was that we were able to provide novel evidence to support this new measure. The psychometric results showed consistency with previous findings of adequate cross-sectional measurement properties. We were able to use the data to address the gap that the FDA had identified in defining meaningful within-person change. There were limitations of the study, symptom worsening, and was generally not observed. And this was across all the PRO measures that were included in the trial. A few participants reported high severity of the symptoms at baseline. And as I mentioned earlier, we had to change the wording of the PGIS and PGIC mid studies given the feedback from the FDA. So this caused a little bit of challenge at the time of interpretation and analysis of the data. This is my last slide, and something else I wanted to just share with all of you, some lessons learned when we decided at Merck to include this a new instrument into our phase 3 study. So prior to the start of the study, we had to get internal concurrence, cross-functional alignment, because we were proposing to include a new PRO instrument in a clinical trial. We had to ensure that we had a licensing agreement in place with the PRO consortium, and also we had to coordinate any translations that we needed with the translation vendor that the PRO consortium works with. And something that obviously we learned is from the beginning, ensure that the wording of the anchors are how we would want them at the end, so to avoid that there is any changes mid-study. During the protocol development, we had to pre-specify points of interest. We pre-specified, administering up to week 18. And this was a little bit of a compromise within our teams. Since all other instruments continue to be administered, we compromised to have that non-small cell lung cancer SAQ and the anchors administered up to week 18. Now, because we had never used, obviously, this instrument at Merck, we had to have new data specifications to ensure that we could include it in the randomized control trial. And we also had to align on the necessary analyses. For this specific study, we had pre-specified analysis plan, and these were conducted by IQVIA. The data sets were not transferred to IQVIA, but IQVIA had to connect to our Merck server. So all of that needed to be planned well in advance to ensure that at the time of database lock, IQVIA had access to our system. And then before and after our interim analysis 1 and database lock, we also had to plan to ensure that we followed the term of the agreement, where it was established that a report was due back to the PRO consortium within 9 months of interim analysis 1 or database lock. So we had to make sure that we had everything in place, to write the report, have internal review done, and then sent to the PRO consortium on time to make sure that we met our agreement. So I hope these were helpful, and I look forward to any questions that might come in the chat later. And at this point, I will turn it over to Margaret.
Thank you so much, Josephine, and hello, everyone. It's really nice to be here and see so many familiar faces on the attendee list. Well, I'm very pleased to present for the final presentation in this session a case study where we used a mixed methods approach, including both anchor-based analyses as well as exit interview approaches, to evaluate meaningful within-person change threshold for the worst itching intensity numerical rating scale within a chronic kidney disease associated pruritus population. But before I go into my content, I do want to make a couple of acknowledgements. So I'd like to acknowledge Cara Therapeutics, who sponsored this program of research throughout, and particularly Fred Menzaghi and Catherine Munera, who were wonderful co-collaborators with this research program. And then a couple of colleagues who I know are in the audience today who are involved in this program over the years, including Maria Matera, Beck Speck, as well as Laura Sweat. So very briefly, I will give a background on chronic kidney disease associated pruritus, talk a little bit about this particular patient-reported outcome instrument, the WI-NRS, and then I'll go into the methods and the results of the various analyses as well as qualitative methods that we use to evaluate meaningful change threshold, talk a little bit about the triangulation of evidence and some of our conclusions, and then a few key takeaways. So pruritus is a common and distressing symptom that's associated with decrements in health-related quality of life among patients with chronic kidney disease. And it affects a majority of patients receiving hemodialysis. And patients who do experience pruritus tend to have moderate to severe symptom. Historically, there was no treatment approved for this particular symptom associated with chronic kidney disease. And Cara Therapeutics recently received approval for Korsuva, which is an injection for treatment of pruritus, particularly in chronic kidney disease among patients receiving hemodialysis. A patient-reported outcome was used as the primary efficacy endpoint in the Korsuva clinical development program. And in particular, you can see here the exact measure that was used, that was also under evaluation in this program to evaluate what a meaningful change threshold would be for this particular item. So the worst itching intensity NRS asked patients to evaluate their itching over the past 24 hours, ranging from 0, no itching, to 10, worst itching imaginable. And I think it's also interesting and important to point out that worst itching intensity NRS items are used very commonly across a number of different therapeutic areas. And when we embarked on this research, there was some literature around evaluation of psychometric properties of this type of item within other therapeutic areas, particularly atopic dermatitis and psoriasis, and also some literature evaluating meaningful change threshold within those dermatological conditions. But at least at that time, there wasn't any literature on evaluation of this particular instrument in a chronic kidney disease patient population. So throughout the clinical development program, Cara Therapeutics had a series of meetings with the FDA, which were very informative to their measurement approach. And in particular, they had actually initially started out by proposing use of a visual analog scale, and through various meetings with the FDA actually did change and switch to an NRS, which was recommended by the agency. And also in a type C guidance meeting in 2015, the agency also recommended that rather than a baseline end of study mean change as the primary analysis, they did recommend a responder analysis to be used as the primary analysis for this clinical development program. So that really made it very important to Cara to consider undertaking some research to hone in on the appropriate meaningful change threshold for this particular patient population with this particular treatment. And then in addition to that, at an end of phase 2 meeting, which took place in 2017, there was some additional discussion around this primary analysis, which was again recommended to be a responder analysis, with the FDA suggesting that they believe that perhaps a meaningful change threshold of 4 points, a 4-point change, would be considered meaningful. And then there was a lot of discussion around different methods that the sponsor could use to continue to investigate and really hone in on whether or not that 4-point change would be most appropriate, or if there was some other level of change that would be considered meaningful for this particular patient population and in this context of use. So we engaged with Cara to develop, again, a program of research to evaluate this question of what an appropriate meaningful change threshold might be for this particular patient population. And that involved a secondary analysis of phase 2 data using anchors of a PGIS and PGIC, and the specific wording and response options for those anchors actually are included on the next slide for those of you who are interested in that. The phase 3 trials did include a PGIC. So really just as a post hoc confirmatory analysis, we replicated the phase 2 analyses using the PGIC in order to ensure that estimates were similar and comparable between the phase 2 and the phase 3. And then finally, we did also undertake phase 3 exit interview study, which included a couple of different approaches. So when we undertook this research back in 2018, exit interviews were really an emerging methodology, and there wasn't particular consensus around the exactly appropriate or most valid, I guess, approach for exit interviews. And so at that time, there was some emerging literature, and so we used 2 different approaches, which involved a modified PGIC approach, and then actual review of patients' WI-NRS change. And I'll talk just a little bit more about those couple of approaches and subsequent slides as I get into the results of those exit interviews. So first, the phase 2 secondary analyses using a PGIC approach as well as a PGIS approach. So results suggested that among those patients who had a minimal improvement based on PGIC, their average WI-NRS scores were around 2.26. I should say average change scores were 2.26. And those who had achieved a much improved change on the PGIC had an average change on their WI-NRS of 3.41, so essentially ranging from 2.3 to 3.4. And then among those patients who improved at least 1 point on the PGIS, and that was a 5-point response scale, their average change on the WI-NRS was about 2.5. And in particular, because that was a categorical change in severity, so for example, patients would have moved from very severe to severe or from severe to moderate. That really was defined based on some clinical expert input as well as some evidence for the literature as probably the best estimation of meaningful change for this patient population. And so we were really looking at something between 2 and 3 points, or maybe between 2 and 3.5 points as meaningful change based on those analyses. Phase 3 analyses were very, very similar. And again, the PGIC was used in the phase 3, and so those were the analyses that we replicated. But among those who had minimal improvement on their PGIC, they averaged less than a 2-point change on their WI-NRS. And for those who had more substantial improvement, so indicated that they were much improved, the average WI-NRS change score was again right around 3.5. So getting into the exit interview methods and then results. So as I mentioned, we utilized 2 methodologies in these exit interviews. In particular, we interviewed approximately 70 patients. We did do stratified recruitment of patients who were exiting the phase 3 trial to make sure that we had a range of patients with different point changes. So we actually targeted having 10 to 12 patients within a 1-point improvement, a 2-point improvement, a 3-point improvement, and a 4-point improvement to make sure that we had, again, a range of actual response within the clinical trial. And then all patients completed a modified patient global impression of change, where they were asked to rate their change in itch during the clinical trial, and patients were able to indicate my itch got worse, no change, my itch got better, but the amount of improvement was not meaningful to me, and then my itch got better and the amount of improvement was meaningful to me. And then after they completed that MPGIC, they actually had the opportunity to view their actual change score. So the interviewer presented them with their data from the clinical trial. So for example, saying, right, so at baseline, you had indicated this as your WI-NRS score, and at end of study, you indicated this, and the change was, for example, 2.4 points. And then patients were given the opportunity to talk a little bit about that change and really what that meant to them and whether or not that was meaningful to them. So in terms of results, very interestingly, the 2 methodologies did yield slightly different results. And the MPGIC results are on the left-hand side of this slide. And then the actual change score results, where patients saw their change and then had a chance to talk through that change, are on the right-hand side. But essentially what you can see here is that anyone who had under a 1-point change or under a 1-point improvement, no patients actually rated that improvement as meaningful. And as you move to the right along the x-axis, for patients who achieved increasingly greater changes in their WI-NRS, increasingly there was a larger proportion of patients who rated that change as meaningful. So again, within, for example, a 1 to 1.99 change on the WI-NRS, about 40% of patients rated that change as meaningful. And then getting into a 2 to 2.99, about 50% of patients rated it as meaningful. And then sort of over that 2-point threshold, a majority of patients considered that amount of change meaningful. And then on the right-hand slide, this was the method where patients actually saw their point change and then were able to talk about whether or not that was meaningful and why, what it really meant to them. And as you can see here, really the majority of patients who had a 2-point or greater change on their actual WI-NRS in the trial considered that change to be meaningful. And there was some very interesting discussion around the benefits that they saw in their lives with that change. So very briefly, triangulation of results, and what our key takeaways were that we had a variety of anchors that we used in the phase 2 analyses. And our primary anchor was that 1-point change on the PGIS, which we considered to be clinically meaningful, given that it was a point change in severity. And that really indicated about a 2.5 point change on the WI-NRS might be meaningful, although certainly there was a range from 2.3 to 3.5. Again, very similar results using the PGIC in the phase 3 analyses. And then in the phase 3 exit interviews, again, the two methods yielded slightly different results, but certainly getting into that 3-point change category, all patients who actually had a 3-point or greater improvement and then had a chance to review those results found that change to be meaningful. So based on all of those results together, from the scientific perspective, we were definitely thinking that perhaps a 3-point change might be considered a meaningful change for this particular patient population. And Carla went on to submit their package and all of those evidence to the FDA, and were successful in their approval. And the WI-NRS now does appear in the Korsuva label. But I think that what's quite interesting here is that the FDA chose to really, again, focus in on that 4-point change improvement. Of course, those analyses were also presented in the evidence package, and the label emphasizes the 4-point change responder analysis and the dissection between treatment and placebo, particularly with that level of response. So a few key takeaways from this presentation. Certainly the come early and come often mantra, I think, came through. I think Carla really benefited from their numerous interactions with the regulatory agency around their endpoints and their endpoint and analysis strategy, and even their strategy for evaluating meaningful change, and certainly took a lot of good feedback and implemented that throughout their clinical development program, and ended up with a successful approval, which is wonderful. Certainly we benefited from having data available in phase 2 and including multiple anchors in the phase 2 so that we could do that secondary analysis prior to phase 3 and really take a deep look on what kinds of change thresholds might be meaningful. We had multiple anchors that we were able to look at ranges of what might be a meaningful response. I think the exit interviews were very illuminating. Actually, in preparation for this presentation, Carla's in the process of developing some other therapeutic areas and other clinical development programs. And I think that they're considering actually going on and doing exit interviews in a number of different therapeutic areas that they're working in, because they found the feedback from patients just really invaluable in a number of different programmatic elements. And then I think also just very interesting from a methodological perspective, there may be benefits and drawbacks of different methods. And in particular, I thought it was quite interesting how the couple of different exit interview methods that we used and based on literature yielded slightly different results. And so I definitely think that that is, fascinating and a topic for future research. Okay. So Bex, I'll turn it back to you to lead us through our discussion.
Yeah, thank you, Margaret, and thank you to all of our presenters. At this point, and we can go back one slide, please. So at this point, I would like to invite our panelists from the FDA to provide their feedback and comment on the content that's been shared.
So good afternoon. Thank you to the organizers for the opportunity to participate on this panel. This is an obvious hot topic. I also want to thank all the presenters for their informative presentations. There was a lot of important as well as interesting information shared, so thank you. I'll be providing the regulatory perspective on clinically meaningful change, and I'll begin my remarks with the Sysaqol IMI update and then move on to the 2 case studies. So I appreciate the efforts of the Sysaqol IMI Consortium. There is definitely regulatory interest in the standardization of data analysis, interpretation, and reporting of COA data, not only in oncology, but in other therapeutic areas as well. And this is evident from the FDA efforts that was described in session 1 with the Standard Core COA grant program, but also with other ongoing data standard activities. The work package 6 objectives are of importance. It will be critical to harmonize the terminology related to clinically meaningful change so we can all speak the same language, right? FDA reviewers see so many different terms related to clinically meaningful change, some of which are not necessarily interchangeable. And from a regulatory standpoint, we are more interested in what constitutes a clinically meaningful within patient change and scores from the patient perspective. It was also nice to see some of the findings from the literature review. It will be interesting to see a summary of the methods utilized for establishment of meaningful change in cancer studies. As mentioned in session 1, anchor-based methods are the primary methods that FDA uses to interpret meaningful within patient score changes and COA endpoint scores. So there is some curiosity, at least on my end, as to what methods are actually being utilized in the studies included in the literature review. And the selected results that were shared from the review of comparative trials, it did show various types of statistical analyses used across several studies, which is not surprising. And it actually mirrors what we see across different medical product applications. While standardization of data analysis will be helpful, it's important to note that the type of data analysis that will be used will be dependent on the COA objective. And this goes for all therapeutic areas, not just particularly for cancer trials, but all therapeutic areas, but particularly for cancer trials, just due to the unique challenges that they face with collecting and interpreting COA data. For example, a COA objective of disease symptom improvement may be challenging to assess if the disease symptoms are similar to the expected treatment-related symptoms or side effects, or if the population is not sufficiently symptomatic at baseline. So evaluating the magnitude of change may not be the ideal outcome for these particular scenarios. Fortunately, our colleagues from the Oncology Center of Excellence have put out a draft guidance, which is entitled Core Patient-Reported Outcomes in Cancer Clinical Trials, which should be helpful for sponsors when formulating their COA objective and measurement strategy for cancer trials. So moving on to the case studies. I have a few general comments before speaking on some of my observations related to these cases. Often a lot of effort goes into the selection and or development of the target COA, and unfortunately the external anchors are left to the wayside, but they also have an important role. They help us to interpret the clinical meaningfulness of the COA endpoint. And in saying this, sponsors should carefully select adequate anchors to conduct their anchor-based analyses to interpret their target COA endpoints. David nicely laid out some considerations for selection of external anchors in session 1, which emphasized the use of interpretable anchors with distinct and non-overlapping anchor response categories, as well as the alignment of the concept and recall period of the anchor with the target COA endpoint to the extent possible. After conducting anchor-based analyses, what we hope to see at the end of the day is that when a sponsor proposes a threshold or a range of clinically meaningful within patient change in their COA endpoint score, is that they're providing an adequate rationale, as well as, of course, evidence to support that threshold or range. And as mentioned earlier in session 1, qualitative methods are viewed as complementary to anchor-based methods. They can provide valuable information, as we've seen with some of the case studies, for some development programs, particularly if anchor-based analyses are not feasible to do or are uninterpretable and or challenging to interpret. For example, if the studies use inadequate anchors or no anchors at all, or if the studies have insufficient sample size, which is commonly seen in rare diseases. So if sponsors are embarking on exploring qualitative methods, we do encourage sponsors to submit their qualitative study protocols, including their interview guides, if applicable, to ensure that these studies are structured to obtain useful information to form a threshold or range of clinically meaningful within patient change. So going on to the case studies, for both case studies, it was nice to see that multiple anchors were used. We generally recommend the use of multiple anchors to provide an accumulation of evidence to help interpret the clinical meaningfulness of the COA endpoint score. At the minimum, we recommend a current state global impression rating scale, such as the PGIS, and a global impression of change scale, such as the PGIC, and we saw both studies use those. From a regulatory view, a current state global impression rating scale is generally the primary anchor, as it allows for baseline and post-baseline assessments, but a global impression of change scale can also be informative. It's just a matter of considering how best to use it, particularly for longer trials. For example, if the studies are 1 year or longer, as it may impact recall. For both case studies, some of the external anchors presented some potentially indistinguishable response categories. For example, there's severe and very severe, much improved and very much improved, et cetera. It's important when selecting response categories for external anchors to be mindful that the response categories are distinct and non-overlapping. This could be done by testing the anchor items, including their response categories in preliminary qualitative work, for example, cognitive interviews. But in the event that a study has used an anchor where the response categories may overlap, one approach for sponsors to explore when conducting their anchor-based analyses is to collapse some of these categories. For example, collapsing severe and very severe, particularly if there is poor distribution of responses across the response categories. This will help facilitate interpretation of these results. However, we would still want to see both uncollapsed and collapsed data. For the chronic kidney disease associated pruritus case study, I did notice that the recall period for the PGIS used in the phase 2 study was the previous 24 hours, which is consistent with the recall period of the target itch numeric rating scale. However, the endpoint was analyzed as a weekly average score. In this instance, to interpret the COA endpoint score, a more appropriate recall period for the anchor scale would have been a 7-day recall period to be consistent with the assessment time period for the COA endpoint. Lastly, I appreciated the example presented on how exit interviews can be used to generate evidence for what constitutes a meaningful score change on an anchor scale, as well as the perception from the subjects whether they experienced a meaningful change. This is one approach to how qualitative data can be leveraged to inform clinically meaningful within patient change. But one point to make is that exit interviews are not a requirement. Some of these qualitative methods can potentially be built into the sponsor's preliminary qualitative work for their COA measurement strategy via cognitive interviews. And so I'll stop there, and that concludes my comments for now, and thank you.
All right. Thank you so much, Selena, for those great comments. And I also want to thank all of our presenters for such informative presentations. So I thought I would offer some general thoughts that would be applicable to all three presentations. First of all, I think it's important to always highlight that the evaluation of clinically meaningful within patient change really pertains to the COA-based endpoint definition. So COA-based endpoints should reflect an aspect of the patient's health that's meaningful from the perspective of patients and or caregivers, and be capable of supporting an inference of treatment effect within the context of the planned clinical trial. In order to help us make regulatory decisions, we need to evaluate and understand how well results of a COA-based endpoint correspond to a treatment benefit that's meaningful to patients. So knowing how the scores produced from a COA relate to patients' experiences is central to interpreting the meaningfulness of results from a COA-based endpoint. So we all know that some COAs might produce scores that are easier to interpret than other COAs in terms of patients' experiences. So how easily one can interpret a COA score will depend on how closely the concept of interest corresponds to the patient's direct experience and how simple or familiar the metric that's used to express the COA scores. So one thing to keep in mind is while we recommend anchor-based methods to evaluate and determine a range of clinical meaningful within patient change in the COA-based endpoint scores, it's not a one-size-fits-all situation. I strongly encourage people to think carefully and consider different approaches, whether it's qualitative, quantitative, or mixed methods approach, and then be sure to select the most appropriate method for your development program. So on this point, FDA is certainly very open to discuss and review different approaches if the approach is well justified within the context of use. Something I've been thinking a lot about is how best to evaluate meaningful change when the COA endpoint is based on a single item COA that measures a single distinct concept of interest that does not overlap with another concept. So for example, if we have a PRO measure that asks patients to report the severity of their abdominal pain on a 4-point ordinal scale of, for instance, non-mild, moderate, severe, and we have qualitative cognitive interview data that provides evidence that patients are comfortable evaluating their symptom severity with the scale, and that the patients view each category, each response category, as corresponding to a meaningful, distinct experience. In this case, the COA scores themselves are directly interpretable in terms of patients' experiences. So my question is, do we still need anchor-based methods for interpretation? So it's something for people to think about. Another scenario where anchor-based methods may not be needed for interpretation is when you have an endpoint defined in terms of, for example, sustained complete symptom resolution. So in this case, the endpoint itself is meaningful as it requires achieving clearly defined and important health states for patients. And depending on the COA objective, we should ensure that patients enrolled in the study have a sufficient level of symptom severity at baseline so that change could be observed. And this point have been highlighted several times by our presenter and also by Selena. Additionally, also depending on the COA objective, it may be important to consider both improvement and deterioration. And based on my experience from looking at different proposals, we often see proposals that include the same threshold for improvement and deterioration. And people need to understand that this assumes that the patients view the degree of improvement and deterioration in a concept of interest as symmetric. And we know that may or may not be the case, depending on the context. And my last point is that whenever possible, data used to inform meaningful change should be different than that used to demonstrate effectiveness. So if you are able to evaluate, explore the meaningful change thresholds prior to starting your registrational trial, because we do have concerns about potential overfitting and generalizability concerns. So with that, I again congratulate to all our presenters for wonderful presentations, and I look forward to more discussion. Thank you.
Okay, thank you very much to our panelists. It's clear that the content is definitely sparking some thought, and we've got questions coming in. Before we start taking those questions, I invite all the presenters and panelists to join me in turning your cameras back on. And I just want to offer the opportunity for you, if there are any questions amongst each other between the panelists and presenters, and give you the opportunity for direct conversation. Okay, well, if not, then we will move on then to opening the floor for the questions. As a reminder, just raise your hand using the icon at the bottom of your screen. I will state your name. You will then be taken off mute and you'll be able to ask your question. Once it's clear that your question has been understood, you can put your hand back down and put yourself back on mute. So to get started, we do have a question from Folka. So Folka, once you're unmuted, please feel free to ask your question.
Thank you very much, Rebecca. So yes, this is Volker again from AstraZeneca. Thank you very much for the presentations. I actually have two questions on the same topic. I hope that's okay. It's on the CMC topic that Johannes presented. The first one is about the term, because I understand that workstream is focused on terminology. So CMC, meaning clinically meaningful change, sounds like it's very focused on change, right? But you, in your slides, you presented to us that you are also looking at something like important differences. Will there be, do you see a problem moving forward with that terminology being, centered around change, when sometimes we're looking at important differences, which may be important differences in group changes, but could be important differences in just their values, based on the landmark analysis that we were talking about earlier. So that's the first part about CMC, directed at Johannes. And then the next one is maybe for Lily Gerard, about the clinical meaningful change again. Same topic. Apologies for my cuckoo clock in the background. I don't own birds. So that's about clinical meaningful change within patients, and whether, if we see in our anchor-based analyses that the results we find are remarkably different across categories of baseline. So what patients perceive as meaningful, that differs greatly depending on where they start. Is that something we should account for in our analysis? Meaning, if we do a responder analysis, should we have a heterogeneous definition of responders based on where you start? Or should we go, this is the average level that a patient needs to change by to feel different. So we go with that for everyone, even though we recognize it's different. So those are my two-part questions. Thank you very much.
So if I may answer to the first question, like the term CMC in our project is basically a placeholder that we use until the final terminology is being developed. And the term itself stems from the grant call that we responded to. And we are referring not only to change in our project, but also differences. And the term change is in that way a bit unfortunate. But as we don't have a better term yet, we're going to use it as a placeholder until terminology has been developed and agreed on. But we're going to focus on group differences cross-sectionally, like changes within patients, but also on group differences in mean change over time, for example. So the focus is definitely wider than the term itself would suggest here. I hope this answers your question.
Yes, thank you very much.
Hi, this is Lily. So thank you for the great question. And I will just speak a little bit about responder analysis and selecting of the cutoff, right? Or clinical meaningful within patient change threshold. So one thing I want to say is that during anchor-based analysis, if you do notice that there is differences across baseline, what patients start at baseline in terms of what they view as meaningful, that may be different. I think that's a valid assumption. So, it's something that I always personally recommend people to take a look, because, this again goes to the argument that it's really hard to select a single cutoff, right? So it's often a range. So if you take, looking at a range, you might be able to incorporate some of those differences from patients' baseline severity status, for example. And then so real quick on responder analysis, and I think, this is, again, it's not a one-size-fits-all situation. I keep saying that because it's true. It really very much depends on the context. So, we should not treat all responder analysis the same. And in certain situations where you have clear evidence to support the meaningfulness of a single cutoff, for example, if you have clear evidence from patients that this amount of reduction is meaningful to them, then in that case, I think, it's reasonable to use responder analysis, with the sufficient and appropriate justification for that. And in another case where if your treatment goal is to reach complete symptom resolution and that the goal itself is achievable, again, using a responder analysis is very reasonable. So I think, people really need to think about their own particular development program and pick an endpoint that is most appropriate. So I would suggest that, if you clearly have uncertainties in terms of what is the exact cutoff, first of all, I wouldn't average, this, I think it just builds to the case where it's really a range. It's not a single cutoff. So you need to think about how you should define your endpoint. I hope that's helpful.
Thank you very much. Very helpful.
Okay, our next question is going to be from Jonathan Gable. Jonathan, when you're unmuted, please feel free to ask your question.
Hello, everyone. This is Jonathan Gable. I work in the digital health oncology group at AstraZeneca. My question is to Josephine, and it's with respect to the psychometric analysis that you performed on Keynote 598. I was interested to see if you conducted any exploratory work looking at individual items, in particular the items related to pulmonary function, so shortness of breath, cough, etc.
Yeah, thanks for the question. So we did do some of those analyses. Of course, with 15 minutes, it's hard to fit everything in, but we did look at, for example, item-to-item correlations and some of the performance of the single items. However, one thing to keep in mind is that the endpoint from the measure itself is the total score, but we do have some of those analyses on the single items, and we have all of that information listed in the paper if you want to have a little bit more information. But the focus then was really on the total score as well. So so we have some, but we didn't do all, you know, and any, like, let's say, you know, thresholds for individual items. That was not part of the activity because the endpoint would be based on the total score. Thank you. If I'm permitted to follow up, this question is more directed towards folks at the FDA. You know, in previous interactions and workshops, you know, representatives from the OCE have referred to their preference for single item assessments, you know, in particular because it aids interpretation. So I'm curious to understand, you know, in using something like a total score using the NSCLC SAQ, you know, how does that lead to any challenges into an interpretation given the multitude of symptoms assessed via the total score?
So I'll start my remarks, and I don't know if Lily wants to add, but I mean, again, this is going to be probably case specific for the development program and the discussions with the agency, right, with the terms of the COA measurement strategy. The core concepts that, you know, our oncology Center of Excellence colleagues do sort of emphasize is, you know, disease-related symptoms, treatment-related symptoms, overall side effect bother. And so depending on what symptom, I guess, that you're trying to go with, it may be worthwhile a single item, but it all depends on, you know, the context of the program itself. So I don't think I can probably give a lot of information without the specifics as to the exact strategy.
And this is Lily again. So I don't think I have much to add on to what Selena just said. I think it's really depending on your program, and, you know, picking the concept of interest it's an important consideration, you know, to embark very beginning of your study, right? So depending on what is considered important to patients to measure and what's the, you know, the overall objective of the study, I think those will be very informative to help you decide, you know, what type of COA measure you should be using.
Great, thank you.
Okay, our next question is going to come from Lindsay Petrenchik.
Okay, so I have a few questions that I can also put in the chat. But one of my most important questions is, how do you choose the time point to evaluate meaningful change? And if you should choose multiple time points to evaluate meaningful change? And also, I've seen a recent paper by Kohler in non-small cell lung cancer that they just used all time points, and I believe a mixed model. So as we're conducting these analyses, it's just kind of hard for us to know which time point we should be using.
So I guess I'll give my comments. I don't know if Lily has anything to add. But I think it all depends on, your product itself and when you're expecting meaningful change. I think that has to sort of inform your strategy. I don't know if there's, a perfect time point, but I think it all depends on your knowledge of your product and when you expect it to change. And I'll stop there.
Do you worry about it being too late, that then it wouldn't represent other people earlier in the trial that have dropped out, if it's like in the case of oncology?
I mean, that is a potential, thing to consider. What is, which patients can variously get there. But again, it's sort of also knowing, when you expect it to change, for your product to have that improvement, and depending on what the concept is as well. So say, for instance, it's something, that may be physical functioning, you maybe need to be a longer trial, right? So it all depends on the concept that you're measuring and when you expect that drug to sort of impact that concept. And those are some things that you need to consider when, I think, selecting the time point. I don't know, Lily,
Okay.
if you have anything else to add.
So I think something to add to what you said is, when do you expect to see when do you expect to see change, right? And I think in a trial setting, you want to measure this, across multiple time points. But it really, in terms of in the end, you want to interpret, that interpretation is based on what your defined, say, primary endpoint is, right? Is it about a change from, for example, change from baseline to a specific landmark, time point, or is it something that's really covering the, entire period of the duration of your trial? So I think, those are some different, you have to think about what your objective is. But I think in general, if you if you have a longer trial, it would be helpful to look at multiple time points.
Okay, and put those all in a model together, or like one longitudinal model, or to do it more descriptively of picking multiple points, like, and evaluating each one of those separately?
So I guess, let me clarify your question. Are you talking about what would be the most appropriate statistical analysis to analyze, like, say, a PRO data, or just about whether it's considered meaningful to patients? Because I think the statistical model would depend on your endpoint, right? So, sometimes we see, MMRM where you include, multiple time points. But I mean, I guess maybe to clarify your question a little bit, because in terms of meaningful change, the anchor-based analysis, we look at the relationship between change from baseline to a specific, depending on how you define your endpoint, and then look at the relationship between the change score with your anchor scale, the categories of your anchor scale. So those are two different type of analysis.
Okay, all right, thank you. And then can we, so if there's a threshold that's derived in, for example, one non-small cell lung cancer population that's, I don't know, maybe stage 4 first, and they're on first line, would that still be applicable to stage 3 second line, or do you have, or should we have specific thresholds for different stages and lines?
Before that question is answered, I'm going to have to say, Lindsay, that's going to have to be your last follow-up, because we have a number of folks with questions as well. So thank you for understanding.
So what I would say is, we would want to provide, a rationale and evidence to support that threshold or range, for whatever new context of use that is. It may be the same, but, you would just have to present, that rationale and evidence to us to review.
Thank you.
Okay, thank you. So our next question is going to be from Steve Huang.
Hi, Steve Huang from Otsuka, Director of Clinical Outcome Assessments in the Global Value and Real Evidence Group. My question is to all the panelists who've conducted the exit interviews. How long after the patient's last visit should the exit interview be conducted? In other words, what's the optimal time period after last visit for capturing rich qualitative input on the patient's trial experience, especially if their exit interviews are part of a standalone protocol and not part of the clinical trial protocol? I'm kind of speaking about saliency versus recency effect here.
Margaret, do you want to take a first stab at that?
Absolutely. Yeah, I was gonna chime in. Yeah, so in our particular case study, because we were using that modified PGIC, where we asked patients about the meaningful change or how meaningful the change was that they experienced in the trial, we knew that we needed to have patients very soon after they were exiting the trial, and so we had a very short window after their last visit. I believe it was within 10 days, but we were really aiming within 5 days, but we had sort of wiggle room. And I feel like I should emphasize, that approach was also incredibly operationally challenging because we had a huge amount of coordination between the various sites that were participating in the exit interviews. And of course, we had some assessments that the patients needed to take, and so we had to get those out sort of in overnight mail before their exit interview, and also to get from the sites and the CRO their actual data, because of course we were trying to recruit patients who had we needed to know what point change they had actually had, because we were trying to kind of stratify recruitment, but also we were using that data in the exit interviews. So I think that kind of the operational challenge cannot be underestimated, but I think it yielded a really interesting data, really fruitful results. So I think it's just more a matter of acknowledging ahead of time perhaps some of the challenge of going into something like that and engaging in careful planning and preparation, and just making sure that there's close communication and collaboration between all of the different stakeholder groups, including the sponsor and the site and the CRO.
Thank you, Margaret. Our next question is going to come from Vivian, and then based on the amount of time we have left, we're going to try to get 2 more in. And Vivian, please limit your question to just a single question. Thanks.
Yeah, so my question is quite simple, actually. Maybe it's for Lily or anyone else. I think you mentioned quickly about instead of the single threshold for responder or non-responder, you mentioned about sustained effect. Do you have any, or I don't know if FDA has any guidance on, obviously you're not looking at a single time point, right? So is there any guidance in terms of, can you use the same type of threshold across multiple time points to assume that it is a sustained effect?
I just want to quickly clarify that when I said sustained, I was using it in the context of like an endpoint that's, saying sustained symptom resolution, meaning that the patients at the end of the trial, for example, they would have no symptom. So in that case, it's important to understand that symptom resolution, in terms of meaningfulness, a lot of times it shouldn't just depend on a single time point, right? So we want to see that effect lasting through, a few time points, for example. But this also depends on, what the treatment is actually able to achieve or not. So, and I think I talked about that in terms of, using that as a responder type of endpoint.
Thank you,
I don't know
Lily.
if I'm answering your question.
Yeah, I guess in the interest of time, let me let someone else ask their question.
All right, thank you. Our next question is going to be from Rita Parna.
Hi, this is Ritu from Merck. I'm a health outcome scientist here at Merck. My question is about clinically meaningful difference in terms of, so more and more oncology research is moving forward in the early stage space. moving forward in the future, do we anticipate or at least want to have different clinically meaningful thresholds for early stage versus late stage patients, because expectations from a patient angle might be different in those two settings?
I don't know, Lily or Selena, do you want to comment on that question?
Can you repeat the question again? I just want to make sure I fully understand it.
Yeah, I was just wondering, it's the clinically meaningful difference thresholds in early stage cancer settings versus late stage settings. I mean, I think currently we use the same sort of thresholds, but as we are talking about enhancing this experience from a patient angle, do we see a situation where we would see different thresholds applied for different settings?
And I believe I think this question was maybe asked a little differently from another person, and I don't know if I maybe responded clearly, but hopefully I'll try again. So for instance, are you asking if a threshold for an early stage setting may be different from a later stage setting?
Yeah, precisely. Yes.
Okay.
And I'm sorry if you have already answered that.
Oh, no, not a problem. I just want to make sure I'm clearly communicating as well. So, I mean, in some instances, the threshold may be the same, but like we mentioned before that, we would want to have a rationale and evidence to support that that threshold is meaningful for both those different settings.
Okay, thank you.
Okay, it looks like we're going to take one final question here. Our final question is going to come from Cheng Long.
Hi, can you hear me?
We can.
Hi, thank you. Hi, my name is Chenglong Han. I'm from Janssen Pharmaceutical PL group. My question is, should the concept measured by the select anchor, like for example, PGRS, be exactly the same as a concept measured by an instrument using, for example, numeric rating scale? Okay, for example, both PGRS as well as the instrument to be evaluated, both measures, for example, pain. Okay, if they should be exactly the same, then I think that it's just a simple conversion from categorical scale to numeric rating scale. It seems that it may not offer any additional value. I would like you to share any thoughts you may have. Maybe Lillian, can you?
I would apologize. Could you just clarify your question, a little bit?
Yeah, okay.
The example that you give specifically.
Okay, I'll give you a specific example. For example, when we measure joint pain in the rheumatoid arthritis, right? I use a numeric rating scale to ask about patients. Now, what's your joint pain, right? From 0 to 10, right? 0, no pain, 10 means worst imagined pain. And then if I want to develop clinical meaningful changes, then I use the anchor PGRS. But in the PGRS, I use the same wording. Now, what's the, for example, if this time, what's the level of your pain, joint pain, right? Then it's a follow-up, ask the same question, then I calculate difference. Okay, come up, no change, 1 level change, 2 level change. So therefore, both anchor as well as my instrument, numeric rating scale, all ask the same question about the joint pain. Okay, my question should be the same, or numeric rating scale I ask about 0 to 10 about your joint pain, but PGRS I may ask about overall severity of your rheumatoid arthritis. So therefore, that's an anchor to measure whether that pain is meaningful in terms of disease, overall disease severity. Did I get my across?
Yeah, so I may also invite Selena to comment on this, but one question I have is, why would we not consider developing skill measuring about joint pain using something with less categories like a VRS? Why do we think a 0 to 10 should be the way to go? Because I think using a 0 to 10 numeric rating scale creates some complexities in terms of, you know, what is really considered a difference, for example, between a score of 2 to 3 or a score of 5 and 6, you know, things like that. So if we look at this, and of course I'm looking at this from an instrument development perspective, right? From the very first step, maybe we should consider evaluating joint pain using a scale with less categories if patients are able to clearly tell you that they are able to distinguish between, for example, you know, severe versus moderate versus mild. Then we could, you know, eliminate some of the challenges with trying to interpret the 0 to 10 scale at the end. That's something that I'm just, first thing that popped up in my mind. I don't know if Selena wants to have some additional input.
Yeah, I mean, it all depends on what scale you're using, right, for the target COA endpoint. If you are choosing to use a 0 to 10 NRS, which has been used with a lot of pain endpoints, right? The challenges that we find, which Lady described, is, you know, how to interpret, you know, a change on an 11-point numeric rating scale. You know, what does going from an 8 to a 7 really mean? And so that's when you have, that's where an anchor comes into use, right? In terms of using sort of maybe that verbal rating scale to sort of help interpret what amount of change would be translated to that NRS.